Okay, so experiment 10 is called "Is this broth culture a pure culture?" And the purpose is to reinforce the ideas of colony morphology and the cell morphology, um, and that they are distinct things. I kind of read the purpose kind of quickly, and so I've like messed up on every single reading of the purpose. But all right, whatever. Anyway, so yeah, that's the important thing. If you just look at the overall scheme of the lab, it's very methodical in taking each step of what you would do, and looking at them very individually. This experiment kind of wraps up the past experiments 5 through 9, in that what it shows you, it reinforces the idea that the colony so you streak something and you get a colony morphology, but then you take the loop, is you take an isolated colony and then you look at it underneath the microscope to get the cell morphology, and that's what this lab is kind of just wrapping up and reviewing those past couple labs. Now it is reinforced here, but stuff like this usually isn't, like the overall scheme usually isn't tested on the midterm. What's usually gone over are very specific things, and it's kind of implicit that you get the overall picture. But yeah, so that's the purpose of this lab, is to do kind of like a review. And so what the procedure was that you were assigned a letter A through H that corresponded to a specific broth culture, and then you did a streak plate of that broth culture, and then you did a simple stain. The simple stain allows you to see the bacteria individually, and the streak plate allows you to see if there are different colony morphologies. And then again, you see the differences here. I'm gonna read y'all something that she gave us, because it can kind of clarify some things. So she said, they do a simple stain on this day and view it underneath the microscope. By considering only the simple stain results, they should answer the question, is this a pure culture? And then they record the answer in their lab manual on page 44. 4 of the cultures are pure cultures, and 4 of the cultures contain 2 different bacteria. One of the mixed cultures contain 2 rod-shaped bacteria. One of the mixed cultures contain 2 coccus-shaped bacteria. Students who get those cultures will likely think that they have a pure culture after viewing the smears, unless they have a good eye for size and tell the rods apart, and cell arrangements to tell the cocci apart. The other 2 cultures are rod-shaped bacteria and a coccus-shaped bacterium. So, what she's kind of getting at is that you want to look very closely at your results that you get, both in the culture and in the colony morphologies, because sometimes it can, just at a glance, it might look like a pure culture, but if you actually look closely, you can see, oh, those are actually 2 different types of, bacillus in that culture. Because if you remember from like last video, you can kind of tell that like you had that Bacillus megatherium, like the mega meant big. They were bigger, like the bacillus were bigger than the smaller, maybe E. coli ones that were a little bit smaller. And so because they're both bacillus shaped and they both look like rods, but the different sizes can tell you that it's not a pure culture. Now this can get kind of tricky, because what if the cell's like growing and you have like a small rod shaped, but so some of them look small, but other ones look big. So it can get kind of tricky in that, but usually we leave them out long enough to where they've grown and then they've reached their maximum potential. But yeah, that's the purpose of this experiment, in that it's kind of just a review. The only thing is, I want to make sure to reemphasize that to do y'all's follow-ups, like on page 44, make sure you get those down and make sure you have those finished, because those are easy points to get before you get into your final. So make sure you get with someone if you don't have them, and record the results.